Hello, everyone. I'm Shigang Yue, and today I'm going to talk about my research in the past few years. And the topic is about bio-inspired vision, and also at the end, I'm going to talk about swarm robots. Okay, let's go back to the. So I think first I would like to thank EU Horizon 2020 project, Step Designer, and also Autocept to give us a generous support. When I first introduce myself, I always use this slide. So, as I said, my name is Shigang, and my PhD is about robotics. And at that time, when I tried to find out how to use vision to guide robots, I found it very difficult to do anything because human, as we know, that animal also, we understand the environment use our vision mainly, but for robots, it's very hard. So my journey is about to find how to understand how the animals understand and use their vision to capture things, and hopefully we can get back to the robots and to an autonomous system. So my method is study the animal or human vision, and we build a model, or you call it neural network, visual neural network. It's biologically inspired, or you can see it biologically plausible with some counterparts in the biological vision system. Then we implement into an embedded system and test it. Finally, we hope to build into a very small chip. In that case, we can assemble into a robot or car or other areas for application. And in the past, we get funding from EU and from other public funding bodies to support our research. I think as a start point, I always show this image. So it's hard to understand the vision of a human because at that time I understand this because it's difficult to do experiment with human, and for animal you have the readily the method you can cut the head of animal and to look at inside, make a recording without much restriction. Of course, we need to mind the ethics as well. So in this picture, so this is the Africa locust. So locust, we know that they always migrate in billions and in swarm, but they are quite hard to hit each other. They always can keep a safe distance between each other when they migrate. So why they can do this? Why they can keep relatively distance without bumping each other? And scientists, I think in the early 70s, scientists in California, they found a special neuron in locust, the head, in this position, for example, here, called LGMD, or lobular giant movement detector. So this special neuron, if you look at in the image like this, LGMD. So this neuron has a very special character. In the early days, when scientists were doing experiment, they found that this neuron responded to movement. So when you put the electrode to this neuron, and when there's movement in the scene, when the animal can see some things moving, and there's spikes. So scientists think, this is the movement detector. Anything moving, this neuron will respond to that. So this is the early days. But later on, scientists find it's not only to respond to movement, but it's specially tuned for clearing. So when there's some object come in a direct clearing course, for example, is approaching from the front to the eye of the animal, so this neuron will generate high frequency spikes like this. So when the object is far away, it's only a spontaneous random spike. And when it gets closer and going to collide with the eye, then there's high frequency spikes. So there's a short wave in recording. So the frequency can be as high as 200 hertz, so 200 times per second. So when the object is receding or away from the eye, or the object is just translating movement from left to right, or just move randomly, there's no big spikes, so it's only respond with object approaching or in a direct clearing course. So scientists gradually they revealed this specific neuron that has its mission is for clearing detection, for emergent clearing detection. So if in reality, if there's a object approaching, or a bird approach the locust, approach the animal, so it will detect by the LGMD, and it will send spikes to the motor neuron, and the animal maybe just hide, maybe just jump, to respond to the predator. Because there's lots of knowledge already know that we can build a model from the photoreceptor to the different layers. We know that so animals' variant system, so this is a schematic illustration, always lay in separated in layers. For example, it has a photoreceptor to capture the light change, and also complex computation here. It has lamina. It believes to be there's lateral inhibition in between, and there's the exchange of information here in the medulla, and LGMD is in the further lobular plate. So the variant system actually is layered. So we build the model also has similar structure, have several layers, the photoreceptor layer, and this is the excitation and inhibition layer and summation layer, sum things together. And we also put a competition layer, G layer, to simulate the dynamic of the information flow in between a group of neurons. Similarly, also with a structure called feed-forward inhibition. This is together the light intensity at the photoreceptor layer, then pass away to the pass down to the LGMD to control the LGMD's fire rate or the behavior. And also feed-forward mediation is gathered information from other places and to mediate the threshold of LGMD. So these are all the structures found in LGMD, the feed-forward inhibition and feed-forward mediation. Although we talk it as a model, actually it's you can also think it's image processing methods. So the photoreceptor layer would be an image, would be a camera capture image, and it just capture what it interested is in the change of light intensity. So light intensity, just to give an example, for example, an object approaching to the eye, then this will be captured at the time t plus 1, the last time, for example, last time step. And so this is now, because it's approaching, become bigger. If, just to give it a little bit further how it works. Then light change or change of illumination means that, it's, the change is just a circle like this. This is the output of photoreceptor layer. So it captures the change, the edge, because it's become bigger and bigger. And if you look at the next layer, inhibition and excitation layer, it separates. So inhibition layer almost the same, excitation layer almost the same. So in this second layer, you can see if the cells are excited or has value, it should be like this. But there's a excitation, it passes to the excitation layer much quicker, and inhibition is a bit slower. So you can see the size of the excitation and inhibition are different. One is big, one a little bit smaller. And so in the summation layer, the two basically in principle it will overlap. We'll add them together, and if add them together, then you can see the inner layer of the excitation has been erased or by the inhibition. If we talk about excitation inhibition, excitation you can think of something positive with positive value. Inhibition, in a simple word, you can think of something with negative value. So if add them together, the positive value will be erased or be cancelled by the negative value. So you can see the excitation become thinner in this case. So this is happening in the S layer. And so if the object stopped without movement, then inhibition layer become bigger and excitation layer stay the same. So inhibition will cancel all the excitation. So in this case, it seems that down to the S layer, this visual neural network becomes computation, and computation can be stopped if there's no movement at all. If there's no movement at all, no motion at all, then there's no excitation inside this neural network. So only if there's something's moving, something's changing, this neural network will respond to that. So here, it can be cancelled by all the inhibition. So in this case, the LGMD neuron or LGMD neural model here we are constructing can capture the things are moving towards the camera or towards the agent or robot if we use the robot camera. So, but it's not to respond to other movements. Here, S layer, G layer is try to enhance the true feature. For example, neighboring cells are all excited, then it will amplified. And for the one that oscillated, excitation will be cancelled because of the distance is travel. It's going to disappear, but it's enhanced the grouped excitation. As I mentioned, feed-forward inhibition is also very important because if you know that a human, we see things, use our eye, and our eyeball capture the interesting things, not in a continuous movement, but our eyeball can jump from one location to another. So in that case, this is called saccadic movement. The eye is saccadic. You move your things, either use your head or eyeball movement. So in the locust, they have a similar strategy, or we believe they have a similar strategy. This is done by the feed-forward inhibition. If things are turning, if the animal just turning, then the LGMD will be shut down by the feed-forward inhibition because there's too many things happening, and this FFI will be excited and is cut this output. So the model, how it works, we explained in on the back, but let's see that its performance. So we run the model, we feed in the model with the images, the stream like this. So you're driving towards another car. Of course, this is a fixed car, it's a balloon car. Then when it's far away, then you can see the output of the LGMD visual neural network or model is quite steady. It's you can call it quiet, but when it gets close to the real object, the car, then the output is climbing up rapidly. And because it's above the threshold, few frames, then it's triggered alarm. If this is a robot or car, is going to trigger the brake. So in that case, the model can be used as an emergency system to detect imminent clearance. So against different noise, the model works pretty good. Yeah. For example, you can see the added noise. You can see its background is quite noisy. But the key feature, the expanding edges are captured. So you can see that all above threshold, even if with different level of noise. This can be directly used in the robots to, we implement to this robot to test our system. So it's like we have a lot of toys and let the robot play with it. And this is Kepler 2 robot we used. Now we have the Curious robots. They are quite small, and the Curious is even smaller than the Kepler 2, not just 4 centimeter in diameter. And the background can be quite noisy, can have some papers, and there's wind also. Also, the lighting condition are quite out of control. You can see there's a strong sunshine there. From the top, you can see this small arena is like this. So the robot can, with the LGMD, with the clear detection with your neural system, it works pretty reliable. So it detects some objects here and is turned to this direction, and detect another object here, then turn to this direction. So it can go on and for a long time. If look at the background, look at the inside the LGMD, you can see the output of LGMD is like this. If it's below threshold, that means it's far away from object. For example, here this period corresponds to this, in this position. And this position corresponds maybe somewhere here. Look at this direction. But when it's close to an object, for example here, then you can see threshold, above threshold, and generate lots of spikes. And this control the robot to make a turn or make a decision. And if you look at the G layer, the last layer before it's summed by the LGMD, you can see the edges are clearly captured. But this method is quite different to the normal computer vision method. You detect edge. We are not interested in edge only. What we are interested is in the expanding edge, how thick the edge is, not 1 pixel, but maybe 10, 3 or 4. The thickness of the edges matters. So that means the expansion rate of the edges are quite important. At different speed, you have different results and behavior. And one important character is that when speed is low, it can get very close to the object. When speed is faster, then it detects the clearance earlier, like this. This is important if you have an autonomous car. If you drive in a high speed, you want to find the threat early to make a brake. So for the driving scenario, we have a database like this, provided by the Volvo Car Corporation. This is in Sweden. This is the driving scenario, and with some pedestrians with roundabout. Of course, roundabout is different to ours, and it works quite fine. And most of the maybe 95% of the scenario, it works perfect. And because it's for the LGMD, the looming sensitive or clear sensitive neural network is from fly insect locust. So it can used by drone or other UAV system in fly. So we put the embedded module the vision module here, a small camera here, and the module we developed to a drone, integrated with the drone, and it can perform like this. So it's fly forward and find the object, then it's turn and get away from the object. So it works perfect for 3D, in a 3D world, in a real world. As I mentioned, we already developed this embedded system. So this is the camera, very small, just 3 gram in weight. And we use ARM chip as a central processor. And this embedded system, it actually is an autonomous robot with vision. So it has not only vision sensor, but also has infrared and other few sensors as well. We have done experiment and with a checkboard, and let robots move towards the checkboard with different speed. The behavior is like this. It's a non-linear behavior. When it at high speed, it detects potential clearance quite early. And if in an arena like this, so there's a camera at top to make a recording and to track the trajectory of robots. And there are also objects. Yeah. So this is the results at different speeds and with different density, the object density. So most of the time it works quite good. This is after a few minutes and make the recording. You can see some of the missed things here. That means not 100% they are reliable. So we can treat this module or the embedded vision system as a neuromorphic vision sensor. It has the ability to compute things very fast. So it doesn't need to have a normal segmentation registration method as a typical computer vision methods. It just compute the things, the edges, and the expanding rate. And we found it at night. It also works. The only thing is that you need to do some pre-processing, and we take inspiration from another group, the Eric Warrant's group in Sweden, Lund University. They are doing the nocturnal insect vision. And the principle is that you take the spatial and temporal summation. You just gathered, for example, the neighboring 9 or 10 by 10 cells value, add them together, so you get 100%, or 9. You get 100 times bigger value, or if you get a 9, you get 9 times greater value. So you make the high resolution to a lower resolution, but in that case you get the spatial summation, and also you need a bit more time to accumulate the photon, the light intensity, and in that case you just wait for 100 milliseconds, for example, add them together, and through this spatial and temporal operation, you can enlarge the value of 100 or 1,000 times. These 3 images are the same that you can't see from the image. But after you add them 100,000 times, you can see the real object. And at nighttime, we want the LGMD, the lumen sensitive, or the depth movement detector works as well. So here, that's the pre-processed method by the PhD student. So here in the A, in the left side, the A, you can see it's roughly there's a plate here, the car register plate. But after pre-processing, you can see almost clearly there are 2 cars. Here, almost you can't see nothing in the C, but after pre-processing, you can see there's a frame on the wall. And for this side, for the left side images, so this is the daytime, or with light switch on. With light switch off, you can't see anything because it's at night and with light, so you can't see anything. But after pre-processing with the nocturnal insects inspired the temporal spatial summation method, or sometimes we simplify it to use log transformation. And you can see the object clearly, and very interesting effect is that not only it can keep the color tune as original, you can compare the original, this are the original images. So almost you can see the green color, the red, and they are almost quite similar to daytime. So that means this method can keep the true color as it is in the daytime. Now with this pre-processing, we can add them with LGMD, and it can detect clearing at night. So that's something we want, because we want the robot to work at daytime and nighttime without interruption. Also, if you think of a robot or vehicle, you want them to work even if in fog or with snow. So in that case, normal camera would work. They can't capture the true scenes, but thermal can still work. And we have a little test. If you feed in with the thermal images, the LGMD, the depth movement detector, the clearing detector can still work. So this, you can see the LGMD can still detect objects with the thermal images. That means, even if in fog, you can still see dangerous pedestrian or a car in front of you. So we talk about the the neural system can detect just one cue, the depth clearing, in depth. But the vision system is complex than that. So it's we found that sometimes we need to detect other movements, not only the depth movement. For example, the driving scenario, there are road marks, and if you're driving, it seems that this arrow become also the movement involved in translating as well. Here, the car in the roundabout, the car is moving from left to right. When your car is moving, if you wait before a roundabout, and also maybe this is truck is seems moving from left to right. So anyway, this involved in other type of movements, not only the motion in depth, but only in left, right, and up, down, in other directions. So we found that it's quite interesting that there are neurons in the insects, and also in high-level animal like rabbit, human, even monkey. We have a special type of neuron that we call it directional selective neurons. They respond only to the movement to certain direction, for example, to left or to right or to something above. So it has this kind of special neuron. We don't know why, but there are neurons in animal. And these are the neurons scientists found in locusts. So these neurons are called directional selective neurons. They respond to different directions. Because we already built the LGMD with the visual system from photoreceptor, different layers down to LGMD, now we can have the similar method to build directional selective neural models. So the only difference is that the inhibition, so excitation is something positive, inhibition is something canceling to negative value. So inhibition arrangement has a special structure only spread to one direction. For example, from here, only spread to the down layers to the left. So all have a similar bias. If they have a biased structure, then it causes the certain type of movement to be cancelled. Here, because it's cancelling to the left, So if something is moving from right to left, yeah, for example, in this case, an object or the stimuli moving to this direction, then when the excitation comes down to this area, it has already been cancelled by the biased inhibition, yeah, because of the biased structure. So in that case, we can build a neural system only respond or only cancelling certain type of directional movements. So we build a 4 neurons example. So left, right, up, down, cancelling neuron system. For this scenario, the pedestrian is running from the right side to the left side. So we know that human movements are quite complex actually. You can't, because the lens, the leg, for example, is not only from left to right, but can go up as well. But the 4 neurons respond in principle, 3 are quite similar, but the 4th one, the left, are different. In that case, with only after a few frames, that means 100 millisecond strong time. After 100 millisecond, so the 4 neurons can tell the difference, can determine, yes, this is it's moving, something moving from right to left. Once you can detect the movement from certain direction to certain direction, you can combine these things to do lots of further processing. And for example, you can build with neural network structure, you can extract specific visual cues. And for me, I think I'm interested in collision, so I can build with the directional selective neuron. I build a collision detector, similar to LGMD, and we can let it evolve, can learn, and to see how it can perform. So this is one evolved structure with directional selective neurons, and it has similar performance like LGMD. And when it gets close to object, it climb up and trigger alarm. Yeah, it can detect clearing 560 milliseconds before clearing, so it's quite good. Still have enough time to take action. And we have talked about the bio-inspired vision system can detect clearing normally in depth and from the front to the camera to twist yourself, and also something moving to left to right, up to down to different directions. And ask myself, so the animals vision system, although we don't know why, but they have this type of different type of specialized neurons. Are there any other type of neurons which can also respond to the primitive motion type? Because I'm a roboticist, I know that we have 6 freedom and X, Y, Z in the 3D world, and also with the rotation, you have 3. So can we find some neurons that also detect rotation? At that time, the answer is no, because I'm in a group study insects, and nobody found that there's a neuron detect rotation in insects. Yeah, but we don't know. Yeah. But luckily, I found that the Japanese scientist, Sakata's group, has already found rotation perceptor or rotational movement detector, I can call it, in monkey. And they put electrode on monkeys' brain regions, and they make recording, and they identified rotational movement detector neurons. So one type is clockwise neuron, and another is anti-clockwise neurons. If you show something like this, an object is clockwise, then clockwise neuron will respond with high outputs, but the anti-clockwise is quiet and without respond to anything. And similarly, you can show in this type, and also object like this, not only bar, but object has a center. As long as it rotate in the clockwise sensitive neurons will respond to that with high output. But if you show some bars moving from right to left, different type, not involved in rotation, so both group are quiet. And so we're quite excited for this, and we want to build a model to show that how it works in principle. We have some ideas because we have directional select neurons. If we can have lots of different directional select neurons, we can detect the directions in sequential way. Just first time it's maybe left, right, then slightly change direction. So if we add them all together, maybe we can, in a sequential way, we can detect it is clockwise or not. And we try this method, and it seems it works. This is the structure of the rotational motion perceptor. So we have the first few layers are quite similar: photoreceptor and excitation inhibition layer and summation layer. And we constructed 16 directional sensitive neurons, in a circle like this. Each respond to interest in a certain direction. And then we build another, next layer, we build another circle. The two circles have biased links. So they all preferred to one direction. So like one, maybe this one's preferred clockwise. So in that case, so the anti-clockwise will be cancelled, but the preferred clockwise will kept and summed in the final neurons, the counterclockwise in this case. And with this model complex, we have from many layers, and we have 16 directional select neurons altogether. So we present them with the similar rotation stimuli, like Sakata's group showed to monkey. And this shows that two clockwise and counterclockwise neurons here, neural models we built. So for this one, the dot just move around its center. So only the counterclockwise respond to that because it's counterclockwise movement. But the clockwise neurons is silent. And this is the bar just moving around center in the counterclockwise way. You can see only the red, the counterclockwise neuron respond to that. And the bar now is clockwise movement around its center. Now you can see the blue one is respond to that. That means the selectivity is quite clear without any ambiguity. So either clockwise or counterclockwise. Now you may ask a question. So if you show some other things, would the rotational perceptor or rotational perception neural network respond to that? The answer is no. And we showed to the rotational perceptor, rotational visual neural network, with the clearancing. For example, the ball is just hit the camera now from far away and just come to the camera. And this is away from the camera. So there's no response to that at all. So this means that there's something like attention, selective attention. So it's only attend to specific type of motion. So that means bio-inspired method is very robust. Okay, I talked a lot about this different type of motion. Actually, it's much complex than we modeled, and we know that also in the same place as LGMD, we have another LGMD2 neurons, and it has similar. They have a similar shape, similar position, but behaved slightly different. LGMD2 only respond to the object, the dark object against the bright background. So you can imagine a locust on the ground when it's young. The most of the things they see is the bright sky, and the bird is something in the dark, like dark object. When they come to the animal, will be a dark object against the bright sky. So when locust is young, the LGMD2 is very important. They can let them to detect the threat and jump away, or find a method to escape from being eaten. So we also build a model. I wouldn't tell the detail, but it's separate the channel to on and off. If you're familiar with the biological variant system, so on means something, the light is become brighter and brighter, from dark to brighter. Is triggered one type of neuron called on response. And off means something from bright to dark. So it's become dim and dim and become dark and dark. So it's will respond by the off cell. So separate on off, you can build this visual neural system only respond to the threat from the sky. Yeah. And with this clearing detection and also directional selective neurons, you can control the robots with the two LGMD or two DSN, directional sensitive neurons. Because you can compare the left and right and to make judgment and to control its movement. Yeah. And I also had asked my question that because we have already find LGMD detect clearing, but we also find directional selective neuron can detect clearing as well, after a bit of a complex neural network. So which one will win? If this, we know that natural selection already make a decision, but in the artificial world, if you have a robot, if you have an agent, so which system you're going to use to detect clearing or detect the approaching objects? So we run artificial evolution, and to detect this, we provide each agent 3 different type of system. We call it the redundant various system. They can use any of them by random or by their wish. So the results show that only LGMD will win eventually in the population. So the bottom are the LGMDs, the upper one are the hybrid or directional selective neuron for clearing detection. So only LGMD win this competition. That means, for the structural reason, LGMD is the natural clearing detector. So another type of visual neural network I would like to introduce to you and draw your attention is called small target motion detector. This has been found in dragonfly. Yeah, dragonfly may be the most complex or most elegant animal that can have the skill and the system that we definitely want to simulate and to reproduce. So the small target motion detector, STMD, they only detect, or they only interest in small objects like this. Here you can see the small black dots. The small target here, we talk about the small object only a few pixels big. So this STMD is just detect that. And against complex background. For example, the background here is forest. So the light condition is very complex. So STMD is for detect small object moving. This you can imagine that dragonfly need to capture the food in this environment. So it must have this ability. To construct this neural network, I wouldn't tell much detail, but it's about separate on and off as well. Yeah, you separate on and off. So something, you have two types of neurons. One is respond to light become stronger. Another one respond to another group respond to light become darker. Yeah. In that case, if one side is triggered, for example, on, and immediately triggered off. If the position of the on-off, the spikes is not far away, that means a small object detected. So principle is like this. First is suddenly become darker, then it's become brighter, or vice versa, suddenly become darker, suddenly become brighter again, or this both are fine. It means that the small object just pass through a certain area. With this principle, we can detect a small object. And we propose another one of the small target movement detector called DSTMD, not only detect where the small moving object is, but also tell which direction it is moving next step. And we have also done something very interesting on ants. So from the nest, this is a part of the investigation of the ants, how they integrate their vision, their memory, and their other abilities altogether. So here we have the path integration, means the ant or the robot can remember its past. So what kind of direction they have moved, and the path, remember the previous path. And also they know some of the landmark, use their vision. And they also have the ability to follow the previous route. So the three optimally combined together, they can always get out, find food, and get back straightforward like a real ants. We have also tried to detect the angular velocity. So why we do this? This is because the bee, when they fly in a tunnel or when they fly in the natural environment, they have the ability to, we believe that they detect the angular velocity and they keep the angular velocity to the same level. In that case, they can follow different terrains and nicely. This is involved in landing control for UAV and also smooth flight as well. So we have the method. Also, we separate on off, and based on that, we detect we have a structure, and we can detect different angular velocity based on the terrain, and to establish the smooth fly method. Another interesting thing I would like to mention is about the simulate the crab's vision system and for detect approaching from different angles. So I wouldn't get the detail on this, but it's very interesting. We had the collaborator in Argentina, and they've been studying the crab for 20 years, and we've been working with them to build this model. To build a model means that we have the neural network, and it can implement into colliers for as generate different behaviors like a crab. And on the way to study these models, we build our robots as well. We call it BioSpide and Swarm robots. The name is colliers. And we have a vision system and neuromorphic sensor, and we have a platform called COS5 and for swarm intelligent research. This is the first generation Curious. I think Fashad, when he was here, I built this tiny robot, and it hasn't got any vision at the time. It only has an infrared sensor and can use infrared sensor to exchange a little bit string and to communicate each other. And now the newest generation, the fourth generation, we have a small camera inside, and it can do lots of things. Still the same size, 4 centimeter. So we have done a few swarm experiments, and this is one of them with different colors. Yeah, it can because we build lots of bio-inspired model. It's very small. The model is very small, very efficient. We can integrate into these small robots without any control. It's autonomous robot. It has its own system, vision system, neural system, and it has different behaviors. Different object coming from different direction shows different intention. It will respond to that differently. Before I finish, I would like to show you some of the swarm experiment. One of the, if you know more or less about swarm robots. So one, of course, this is a very complex area, and so one early experiment is about follow the leader. So we have a leader, when the leader is moving around, it's lay the pheromone, and the other robots, when they detect pheromone, then they follow the pheromone, try to examine the behavior, to examine the behavior, follow the leader. Let's just see that how it works. Just see. So this is a platform we developed in Lincoln, and so we use the early. This is the early generation of robots. So the leaders, the pheromone actually is simulated with on a TV screen. So because there's a camera above the platform, it can detect where is the position of the leader, then it can project the pheromone after the leader. So this is 2 followers. Sometimes they can have issue, but yeah, generally it shows that it can show the follow the leader after a while. Yeah. So the pheromone it laid will evaporate after 10 seconds. It becomes weaker after a while. On the back, it has the code, C code to and Python as well to control all the system. So this is 5 followers for 1 leader. I can see that the swarm robot, we understand very little about the swarm behavior. The previous swarm research focused on the emergent behavior, like many emergent show something amazing, but I think in reality it's much complex than that. And I think vision hasn't been investigated what kind of rules for the swarm behavior. And we have a vision sensor, we have the ability to use vision to study swarm behavior as well. So let's show you the application of the swarm robots can actually be used for other experiments as well. Okay, just see that. This is the newest generation of Curious, and we use it for not only follow the leader, but we used it for the multi-pheromone research. Multi-pheromone means not only one follow leader pheromone, but also expel and other type of pheromones. They may use different pheromones to achieve efficient behavior. And also the right side is showing we want to do the transportation simulation. So this is one of the early stage experiment. On the TV screen shows the car, the vehicle, simulates the vehicle behavior. Yeah. Okay, so all these things are from the researchers, postdoc, and the PhD students in Lincoln and in other teams and collaborators. So especially thanks to Claire and Yoshifumi's group, I think that we've been supported by, from FP6 to 7 and Horizon 2020. And also thanks for the colleagues in Lincoln. I just show one of the project technical meeting here. So we have been in Cambridge just before the pandemic. And I remember that time pandemic just began, we were just worried about if we need to meet or not. Anyway, we have met and very nice workshop there. And yeah, we been eating in the restaurant, but we have some difficulties because of the pandemic. I hope that we can resume the activities quite soon. Okay, I finished my talk. I think I'm talking about the motion sensitive systems in terms of the biological vision and how to model it and what we're going to use. And I show that it can be used in at least in a robotic system. And at the same time, the small robot, we can use it to study swarms. And because it has vision, it will help us to understand how vision can play a role in swarm intelligence. Okay. If you have any questions, you're free to ask, please. Yeah. I'll just get back to the StreamYard. Thank you.